Okay, welcome everyone, and this is the second video for our biology 2. And we started off with talking about proteins, particularly the amino acid monomers and polypeptides. And now we proceed with another video, or the second video for the third quarter. Let's continue discussing about proteins, and particularly let's start off with showing you three pictures wherein I'm going to show you something or a picture of something, and then you will tell me what do you need. So for this picture, what do you need? Of course, you need a hammer. Second picture, what do you need? Of course, it will be a screwdriver. And finally, what do you need for this? It's called a bolt, so you need a wrench. So all of this stuff that you have seen a while ago, of course, they have a particular common denominator, a common thread among these three, and that would be you can only use a certain tool because of its structure. So you cannot use, for example, over here, you cannot use a hammer in order for you to drive in a screw, or you cannot use a screwdriver in order for you to move the bolt. So the structure will eventually determine what we call as the function. So that is a very important thing that we have been repeating over and over again in the past few meetings, that the structure determines indeed the function. And this is not only the case of biomolecules, we have studied that a while back, but it's also very important for macrostructures as well. So for example, our ears. Our ears has a certain structure. It's shaped like a dish, and its function is to receive sounds from the environment, and the structure actually enables the ear to do that. It's able to collect the sounds that will be coming from the environment. So all over again, the structure determines the functions. And with that, we can now go to the protein structure and function, which is the title of module 1.2. And we have the following objectives for this module. Number one is for us to identify the 4 levels of protein. So you have to remember these 4 levels. And number two objective is for us to discuss how the level of protein structure are achieved and the significance of this structure to protein function. So you will be studying in this module not only the different levels of the protein structure, but the accompanying function and what causes this function in these different levels of protein structure. So let's begin our discussion from here. Okay, first of all, we have to know the difference between the polypeptide and a protein. So if I'm gonna ask you, what do you think is the difference between polypeptide and protein? What would be the answer? Well, to help you with that, here are the pictures. So this is what the polypeptide looks like, and this is what the protein looks like. So if your answer would be a polypeptide is simpler compared to the protein, that's correct. If you say that it seems to be a string of amino acid for the polypeptide, while the protein has folds, coils, and that is also correct. And with those put together, the polypeptide is simply a chain of amino acids, simply the sequence of the amino acid, the string of amino acids. That's called the polypeptide, while the protein would be called a complex molecule made up of folded and coiled polypeptides. And take note of this very important characteristic. It is the protein that will become functional and also active. So this means the polypeptide is not yet active, not yet functional, while the protein would be the one that will be the one functioning, or will be the one that will be considered as the active molecule. Sometimes people would interchange these two words, but this will be the difference between them. The polypeptide is a simple chain of amino acid, while the protein is complex, folded, and coiled, and becomes functional active molecule. With that, let's now go to this question. What are the 4 levels of protein structure? So these are the 4 important things we have to remember. The first one is called the primary structure. So if you know the primary structure, it's very easy now to know the other 3 levels of protein structure. Starting with primary structure, you next have the secondary structure. The third would be the tertiary structure, and finally the fourth would be the quaternary structure. So knowing now the four levels of proteins, let's go now into the details of each one of them. Starting with the primary structure, the primary protein structure would simply be the order or sequence of amino acids. It would be the string of amino acids, the sequence of amino acids, the order of amino acids, as you can see all over here in the picture below. So that's simply it. One characteristic of the primary structure is that it is formed by the formation of covalent bonds between the amine and the carboxyl of adjacent amino acids. So you can only form a polypeptide when a peptide bond is formed, and that primary structure will be formed because of that. In addition to this, one important thing or principle you have to remember is that the primary structure will be controlling all subsequent levels of protein organization. So the next coming level, the secondary, the tertiary, and the quaternary structure are all dependent on the primary structure. So here's a question: what if you change one of the amino acids in the primary structure? We'll be dealing with that in the near the end of our discussion. But just take note that the primary structure is the order or sequence of amino acid. With that, let's now go to the second one. We're done with the primary. What's the secondary structure? Before that, we have to take note of this first of all. Another important thing about the primary structure is that there is what we call as the amino end, which is the N terminus, and the carboxyl end, which is the C terminus. The amino acid in this end of the polypeptide will have an exposed nitrogen and hydrogen called the amino end, while the other end will have an amino acid with the COO that is part of the COOH or the carboxyl end. This would be the end that will be exposed here. And you call the amino end as the N terminus, while the carboxyl end will be called as the C terminus. Okay, so we're done with the primary structure. The next question is, what is the secondary structure? Well, the secondary structure, this is where the foldings will be happening right now. So as you can see here, this is a special polypeptide fold, and this happens in a repeating arrangement, and this will be formed two important forms, which would be alpha helices and also what we call beta plated sheets. So the alpha helix over here is colored purple in the picture. You can see here that it's going round and we're swirling round and round, and this is what you call as alpha helix. While on the other hand, you have here side by side the parts of the polypeptide chain that will have interaction, forming a beta pleated sheet, just like a zigzag of formation of the polypeptide, which is symbolized here by an arrow going in opposite directions. So this would be a result of what we call as hydrogen bonding among the amine and carboxyl groups of non-adjacent amino acid, which forms the swirls and which forms the beta plated sheets. So that would be the secondary structure. This is the first of the three-dimensional structure that happens after the primary structure, or simply the sequence of amino acids. So take note of this one. Secondary structure provides the polypeptide chain a level of mechanical stability. It's make it more stable. So we're done with the primary structure and we're done with the secondary structure. The next one is the tertiary structures, and the tertiary structure would be made up of further folding, further three-dimensional structure. So it's a complex molecular shape, a three-dimensional structure of the polypeptide. Now what is happening here, just like in the formation of the alpha helices and also the beta plated sheets, there will also be interactions, but not simply hydrogen bonding only. There will also be disulfide bridges, there will be ionic bonds, hydrophobic interactions, and all of this interaction makes different types of turns and coils, forming together the several alpha helices and several beta plated sheets together into a three-dimensional conformation, and that makes up the tertiary structure. So a few things we have to remember for here is that relative amino acid positions are very important because non-polar amino acids usually avoid exposure to the aqueous solution. So if it's in water, the hydrophobic side chains will usually be found found inside of the complex molecular shape, and that will give a distinct tertiary structure for that particular polypeptide. So again, the tertiary structure may be important for the function of the protein because there may be some parts of the three-dimensional structure of this polypeptide that would be important for enzymes, to act as enzyme, like active site in the enzymes. I will be talking about that later in module 1.3. So you will see here several hydrogen bonds, hydrophobic interaction, disulfide bridges, ionic bonds, and this will be very helpful in forming the final three-dimensional shape or conformation of your tertiary structure. So we have the primary structure, which simply is the string or the sequence or order of amino acids. We have the secondary structure, which would contain the alpha helices and or the beta plated sheets. So this will be a further conformation. And we have now the tertiary conformation, which is more complex. It's a three-dimensional structure of the polypeptide. So we're done with the first, the second, the primary, the secondary, and the tertiary structure. How about the quaternary structure? Well, the quaternary structure will be more complex because instead of having only one polypeptide chain, which is the tertiary structure, you will now have multiple polypeptides or prosthetic groups that interact together. So you have here multiple polypeptides now forming the quaternary structure. So in this case, the very difference between the tertiary and the quaternary would be the number of polypeptides present. For the quaternary, there are two or more, while the tertiary will be containing only one polypeptide chain, and that will be it. And quaternary structure may be held together again by variety of bonds that are similar to the tertiary structure. So putting them all together, we now have this summary. So this is the primary structure, a sequence of the amino acids. We have the secondary structure, further three-dimensional structure here, but you have the alpha helix, or the other one is the beta plated sheets. And then the tertiary structure will contain all of the helices and all of the beta plated sheets together, forming a three-dimensional structure of the polypeptide chain. But the quaternary will now be made up of not only one but two or more assembled units of polypeptide chains. So this would be the summary of the four levels. Here's another picture of that, particularly showing the alpha helices and the beta sheets. And now you know the four levels of protein structure, and that answers the first question. And now we have also achieved the second objective, because we now know how these different levels of structures of protein have been achieved, as you can see over here. So let's now answer some concept check. Number one, what happens if you change one amino acid within the primary structure of a protein, which we asked earlier? So what will happen if you just change only one? Well, the answer to that is, even if one amino acid is changed, it is possible that the three-dimensional structure of the protein can change, thus changing its function. So changing one amino acid has a great impact. It may change the secondary, the tertiary, and even to the quaternary structure of the protein. So for example, if you have an amino acid in the very middle, which would be causing the polypeptide to bend towards the right, and this amino acid was changed so that the polypeptide now bends towards the left, then we can expect that there will be changes in the higher levels of protein structure. One example of this is the sickle cell anemia, wherein one hydrophilic amino acid is changed to a hydrophobic amino acid, which causes the clumping together of the hemoglobin, which eventually changed the shape. And here's a picture of that. So in this case, if you look at the primary structure, the first, second, third, fourth, fifth, sixth, on the sixth position, the glutamine, which is a hydrophilic amino acid, was changed to valine here in the sickle cell anemia, into a valine, which is a hydrophobic amino acid. Looking at the normal hemoglobin with the normal glutamine on the sixth position, you will see that the normal beta subunit would look like this. And then the quaternary structure, where it binds with normal alpha hemoglobin, they will be separated from other such hemoglobin. So the normal hemoglobin proteins do not associate with each other or one another, and thereby carries only oxygen. And because of that, there is no change in the overall shape of the cell membrane of the red blood cell. So this would be because of the individual hemoglobin proteins that are not congregating or are not associating with each other. But the problem with the sickle cell hemoglobin, the change in the 6th position to valine, a hydrophobic amino acid, causes the beta subunit to also change drastically. And when this binds or associate with the normal alpha helix, alpha hemoglobin, it will now cause the hemoglobin molecule or the hemoglobin protein to associate together, and the hydrophobic interaction between these sickle cell hemoglobin proteins would lead to aggregation, and this aggregation produces fibers. So this not only lessens the carrying capacity of the hemoglobin, so it lessens oxygen being carried, but it also changes the shape of the red blood cell, causing it to become sickle shape, deforming it because of the association of the fibers, or the fibers being produced of the association due to the abnormal sickle cell hemoglobin. So just remember, one change of the amino acid could have a drastic effect on the secondary, tertiary, and also quaternary structure. And you will be noticing here that since an amino acid was changed, this means that a change happened in the RNA that helped the production of this amino acid or production of this polypeptide, and the RNA eventually received the change from a change in the DNA. We'll be talking about that in the next coming topics, but just take note of this. One change of amino acid can have a drastic effect on the other higher levels of protein structure. Another question: why do you think only some proteins have quaternary structure? Well, the reason for that is because, first of all proteins would have primary, secondary, and tertiary structure, but not all proteins would have a quaternary structure. The reason being, there are some proteins that are already functional, being only a single polypeptide. You don't need an addition of other polypeptides to make them active, to make them functional. So it only stays at the quaternary structure. However, there are some proteins that in order to become functional, two or more of these polypeptides need to attach together, need to be arranged together so that it becomes functional. So the foldings of protein is also driven and reinforced by the formation of many bonds between different parts of the chain and also different polypeptides producing the quaternary structure. So here are some examples. This is a tertiary structure of the lysozyme enzyme. Only tertiary because it's only made up of one polypeptide chain. Another one is starch synthase enzyme, as shown by this very, very complex ribbon diagram, where you can see a lot of helices over here and a lot of beta-plated sheets shown by the arrows here. This is also only up to the tertiary structure because it's only one polypeptide. You will notice here the N-terminal and the C-terminal over here. Here's an example of quaternary structure, which is again hemoglobin, and in this case it contains 4 units, 4 polypeptides: the yellow, the second is the red, the third is the violet, and the fourth would be the blue. So it reaches up to the quaternary structure because it's made up of many polypeptides together. Another question: what condition affect protein structure? Well, there can be several conditions that can affect the protein structure. Particularly, it would be physical or chemical in nature. So for example, changing the pH can change the protein structure. Changing the salt concentration, too much salt or less salt, there will be a change in the protein structure. Higher the temperature or lower the temperature, protein structure will be affected as well. And there are several other conditions wherein a protein can unravel. And when the protein unravels, or what we call, there will be a loss of protein's native structure, native 3-dimensional conformation or structure. This process is called denaturation. And when denaturation happens, the protein becomes unraveled, and when it becomes unraveled, the denatured protein is no longer active. It becomes biologically inactive. It is unable to function anymore because it is unraveled. So here's a picture of that. So if this is the normal protein's three-dimensional conformation, the native conformation, when there's denaturation, it now becomes unraveled. The three-dimensional structure is lost. So there is a loss of biological activity. So it's no longer functional when the protein is denatured. However, there will be some circumstances wherein just removing the abnormal condition can cause the protein to renature again. So there's renaturation, wherein it regains its normal three-dimensional conformation, which implies a regaining of the activity or the function of the protein. However, there will be some cases wherein the denatured protein no longer reverts back to the normal protein conformation. So it's called to be irreversible. It's no longer reversible. One example of that is cooking eggs. So when you first crack an egg, you will see that the white would be almost transparent. It will be transparent. That you can, it contains albumin and several other proteins. But once you cook it, this albumin becomes denatured protein, even gets destroyed, and it can no longer return to its normal protein conformation. And in that case, the denaturation, when the egg is cooked, is already irreversible. So that would be the whole of our discussion for Module 1.2, and we'll see each other in the next video.